walled off from the mom.
All the nutrients baby needed had to already be in the egg from the get-go. If you think about a chicken egg, that's exactly what the yolk is. It's a care package that has to last the embryo until it's full.
The evolution of placenta essentially involved losing that eggshell and instead replacing that with some kind of tissue or organ that attaches to the mother's uterus.
But losing that shell presents some challenges.
At the same time, it's important that the maternal and fetal bloodstream remain separate. And so the separation of these bloodstreams is established through this cell layer called the syncytial trophoblast.
The syncytial trophoblast is the outermost layer of the placenta, the part that's pressed against the mom's uterus. It's literally a layer of cells that have fused together, forming a wall.
This is where the magic happens. This wall of cells keeps mom and baby working in harmony and not killing each other. There's no other structure like this anywhere else in the body.
When evolutionary biologists like Ed Chung mapped the genomes of these cells, they found something very odd.
So the protein that allowed these cells to fuse into a wall, it's called syncytin, it didn't look like it came from human DNA.
What did it look like?
It looked more like, drumroll please, HIV.
What?
Yeah, what Ed Chung told me is that this protein actually came from an ancient retrovirus, quite similar to HIV.
So you're saying our placentas are made, at least kind of, partially made from viral DNA?
Exactly.
Let's take a step back. Viruses are little strips of biologic information that can't do anything on their own. So they enter cells and hijack the cell's machinery to make copies of themselves and replicate and move on to infect other cells. Retroviruses take it to the next level because they are strips of DNA that enter cells and just go ahead and insert themselves into the host's own DNA. Now the host is stuck with viral DNA for the rest of its life, and it can never stop doing what the virus wants.
Today we have the most famous retrovirus is of course HIV, but retroviruses like HIV have been infecting vertebrates for probably a couple 100 million years.
So the consensus among evolutionary biologists is that once upon a time, some retrovirus infected an egg-laying vertebrate, and by chance, that virus settled into that animal's egg cells.
And it just so happened that that particular infected egg met a nice sperm and got fertilized.
The baby that was hatched, whatever kind of proto-mammal it was, now had copies of that virus's DNA in all of its cells.
Right, because if the retrovirus managed to insert itself into the first cell, it would be copied into every subsequent cell.
Yep.
The virus didn't kill the baby. If it had, we wouldn't be sitting here as humans telling the story. What it did do was give this offspring a premium feature.
So we got an upgrade, like viral status. Viruses fuse with things in order to infect them, right? So now we get this viral DNA that lets us make a protein that fuses things.
This once viral protein essentially slowly morphed or evolved into what we now know as syncytin.
So back to the proto-mammal. It contains viral DNA that codes for this syncytin protein. This protein gives the baby the ability to fuse cells into a wall. A wall that connects mom and baby, but also keeps them separate.
It gives this baby the ability to create a placenta.
Basically, this virus helped that mammalian ancestor survive better by giving it a better placenta. And this piece of DNA would have been passed on to the generations and eventually spread into the population. And in this process is essentially what we think of as a molecular domestication of an ancient retrovirus.
Wow, so the virus tried to hijack us, but we hijacked it.
Yes. But it's worth noting that this probably didn't happen overnight. This was the key step, but it probably took many more generations worth of mutations.